The last thing that we're going to discuss is the environment's effect on phenotype. So we've talked about the environment's effect on phenotype a little earlier when we talked about human skin color. We did talk about how the human skin color is a polygenic trait, meaning that there are 3 genes, and whenever we have multiple genes that control a trait, we call that a polygenic trait. But we also said that the environment, the amount of UV light exposure, can affect human skin coloration as well. So we do know that the environment does play a factor into some phenotypes. Here's some other examples that I see very often on the AP biology exam, and one of those is the effect of soil pH on hydrangea flowers. So notice that when you have a very acidic pH, and remember that these small numbers are acids, and then here we have something that is alkaline or basic at a higher pH. When we have a low pH, these plants can uptake lots of aluminum cations. So the low pH of the soil frees up these aluminum cations, and plants can uptake these through their roots, and the result of that is this deeper blue coloration. When pH is alkaline, meaning it is basic, we do not have a lot of aluminum cations because aluminum is going to be bound to something else and we won't have those free cations. So instead we will have this deeper pink color. So altering the amount of hydrogen ions, which is pH, can affect the coloration of these flowers because it alters how much aluminum they can uptake. Another example that we will see is what I like to call like what went wrong at Jurassic Park. So in Jurassic Park they were supposed to make all of the brand new dinosaurs the same sex so that they would not interbreed with one another. So we had geneticists who really didn't understand sex determination well, because sex determination in reptiles can be determined by 2 different things. So the first thing that can determine sex in reptiles is just their genotype, and that's what we know. We know that the combination of alleles can determine sex, someone's genotype. However, in reptiles, often we're going to see different chromosomes. We won't see X's and Y's, but we'll see chromosomes that are Z's and W's, and we'll see those in some bird species as well. We do know that birds and reptiles are closely related. So the difference here is if we are homozygous, so we have ZZ as a genotype, that would be a male in a reptile or a bird, and then the female is going to be the heterozygote here. So the females would be ZW. So you may see multiple different types of inheritance patterns on the AP Bio exam. The only one that you need to know for sure is this XY system that is found in human beings. If they give you something like the ZY system that we just talked about in birds or reptiles, or if they give you this XO system in insects, or this haplodiploid thing in honeybees, they will explain this inheritance pattern. I will tell you that the only 2 that I have seen on the AP Bio exam have been the XY system in humans and the ZY system in birds. So we're only going to discuss those 2. And I know that if they give you C or D, that they're going to explain that to you when you would be able to figure that one out. So we now know how sex is determined in reptiles by the use of their genes, but we really haven't understood what went wrong at Jurassic Park, because those geneticists determined and they made sure that all of those dinosaurs were all the same sex. So what went wrong if all of their genes were manipulated correctly? Here's the second thing that impacts reptile sex determination. That is temperature. So let's look at these different reptiles. Notice here that it really does not matter with these reptiles what their genotype is at all. So we're going to look at turtles and notice here that their genotype, everything is X. They all have X chromosomes. So what is really determining whether or not they are male or female is the temperature of the embryo during critical stages of development. So over here we have the percentage of males, and notice that if we have a temperature here that's between 26 and what is that, like close to 28 or so degrees Celsius, we're going to have a high probability, look, almost 100% of those offspring that are going to be male. However, during embryonic development, when those turtles are in those eggs still, if that temperature gets high, if it gets to be 29 degrees, then we're going to see a big decrease in the percentage of offspring that are going to be males. Many more are going to be females until you get to 31 degrees right here, and all of those baby turtle eggs will hatch and they will be female. So the main consideration on sex determination in turtles is going to be the temperature during that embryonic period when they're in their egg. So we'll see that in different turtles. We also see this in alligators. Notice that we have different temperature ranges here for alligators, but it works the same way. Depending on the type of temperature you will have either males or females in their clutch. So that explains what geneticists could have gotten wrong in Jurassic Park. So let's say that they just decided that they wanted to make all those dinosaurs female in Jurassic Park, and every single dinosaur that they genetically engineered, they made sure that they had the genotype ZW, ZW, ZW. But some organisms use both the genome and temperature to determine sex. So look here at this 3-line stink lizard. So notice that even if their genotype is XY, temperature can override that genotype and change the sex of that offspring. We see that here with the bearded dragon as well. Notice that this bearded dragon is ZW, should be female, but if the temperature is not at that critical area, the temperature overrides the genotype. So this really explains what went wrong at Jurassic Park. If your genotype codes for female, but the temperature development could be wrong, then those organisms will still develop as males. Here is one more example that I see quite a bit on the AP Bio exam on how the environment affects phenotype. This is the same rabbit. This is the Arctic hare. We see that rabbit in the winter time, and then we see that rabbit in the summer. And it is the same exact rabbit. What is going on with their fur is their photoreceptors. So photoreceptors are here in the eye of those Arctic hares. We have longer days in the summer, so photoreceptors will receive information that there are longer days in the summer and then shorter days in the winter. And depending on the amount of light that those photoreceptors receive, cells communicate to each other, and then different genes are turned on and off. So in the summer, when we have lots of light, we will turn genes for pigmentation on, and we will have coloration of those hairs. When days in the winter are short, those genes are turned off, and then we have this light coloration of fur. So the genes are not changing in this Arctic hare, but what is changing is whether or not that gene is silenced or that gene is turned on and is going through transcription. And it is the amount of light and those photoreceptors that receive that light that is the stimuli for that gene, whether or not it goes through transcription or not. So those are our mostly the environmental effects that I see most often on the AP Bio exam. And there will be a practice FRQ for you to complete. That practice FRQ does have to do with the sex determination in reptiles. I think it's on fish. So if you have any problems with that question, come back and look at sex determination again.